Hello students, today once again we are studying biology class 11th chapter number 7 cell division. We have already started with this chapter. In our last lectures we have studied so many topics related to this chapter. Okay, today we will continue this chapter. In our last lecture we have studied about the topic of meiosis. What do you mean by meiosis? Let us revise the definition of meiosis. It is the cell division by which gametes with half the number of chromosomes are produced. So what is meiosis? The cell division by which gametes with half the number of chromosomes are produced. This term was coined by J.B. Farmer in the year 1905. We have studied in detail about the meiosis. Then we have seen that meiosis, there are two types of meiosis, that is meiosis 1 and meiosis 2. We have studied in detail about the phases of meiosis 1, that is prophase 1, metaphase 1, anaphase 1, telophase 1. In that we have studied that prophase 1 is subdivided into 5 subphases, that is leptotene, zygotene, pachytene, diplotene and diakinesis. So we have studied about the 5 subphases and we have studied about the phases of meiosis 1 in detail in our last lectures. Even we started with meiosis 2 and we have seen that meiosis 2, what is meiosis 2? The two cells produced by meiosis 1 now enter the second meiotic division, and this second meiotic division is known as meiosis 2. Unlike meiosis 1, neither cell goes through a chromosome replication. Each of the cells of the chromosomes has 2 chromatids. Then we have seen about the phases of meiosis 2, that is prophase 2, metaphase 2, anaphase 2, telophase 2. All the phases are similar to mitosis. All these phases are similar to mitosis. And we have completed till telophase 2. So today let us start with the next phase after telophase 2, that is cytokinesis. There are two phases, that is karyokinesis, cytokinesis. In karyokinesis, telophase 1, in karyokinesis, meiosis 2, metaphase 2, anaphase 2 and telophase 2 occurs. And then occurs the cytokinesis phase. So let us see what is cytokinesis 2. Cytokinesis occurs after nuclear division, as I have told you. So 2 haploid cells are formed from each haploid cell. Thus in all 4 haploid daughter cells are formed. These cells undergo further changes to develop into gametes. What is cytokinesis 2? What happens here? It occurs after nuclear division as usual. 2 haploid cells are formed from each haploid cell. Thus, in all 4 haploid daughter cells are formed. These cells undergo further changes to develop into gametes. Now let us see the significance of meiosis as we have completed the topic of meiosis. Meiosis 1, meiosis 2, both the topics we have done. So today let us see the significance part of meiosis. Genetic variations caused by the possible combinations of chromosomes during crossing over in meiosis 1. Crossing over increases genetic diversity as the genes on the chromosomes combine in number of ways. As we have discussed this, that crossing over increases the genetic diversity. There is changes in the gene frequency, as the combination of chromosomes are going to take place in different ways. An alteration in the DNA structure during meiosis occurs mutation, that means sudden change. In most cases, mutations are maladaptive and then the new cell dies. A small number of mutations are viable The cell survives in the mutant gametes, combines with the another gametes to produce an offspring. The mutation can be passed into the next generation. Let us see the functions of meiosis. First one is the production of haploid gamete, so that the fertilization restores the normal somatic chromosomal number. Second is the production of tremendous amounts of genetic variation. Third is segregation of the two alleles of each gene. This takes place due to pairing between the two homologs of each chromosome and their separation at the first anaphase. Next function of meiosis is recombination between linked genes due to the crossing over during Pachytene stage. Meiosis facilitates segregation and independent assortment of chromosomes and genes. Next significance is in sexually reproducing species, meiosis is essential for the continuity of generation. Because meiosis results in the formation of male and female gametes and union of each gametes leads to the development of zygotes and thereby new individual. Okay, so all these are the significance of meiosis, uses of meiosis. Okay, uses, significance, application, all the terms means one and the same. You can write for any of the question, you can write this answer, that is significance of meiosis or benefits of meiosis, uses of meiosis. Okay. Moving further, since we have completed the whole chapter of mitosis and meiosis, there are many points in our mind that shows distinction point between the mitosis and meiosis. So now we are going to discuss the differentiate between mitosis and meiosis. This is very important question. Students mark it important from exam point of view. Distinguish between mitosis and meiosis. What is mitosis? It occurs in somatic cells. Means it occurs in all the body cells except reproductive cells. Meiosis, it occurs in reproductive cells. Means meiosis takes place only in reproductive cells. Okay. Mitosis takes place in two daughter cells and one cell produces two daughter cells, and here in meiosis one cell produces four daughter cells. Okay, mitosis occurs in somatic cells, meiosis occurs in reproductive cell. In mitosis one cell produces two daughter cells, in meiosis it produces only four daughter cells. It is a sequential division separating sister chromatids. Okay, in what is mitosis? It is a sequential divisions separating sister chromatids. Whereas in meiosis, it is a reduction division. The first stage is the reduction division, which separates homologous chromosomes at first anaphase. Sister chromatids separate in an equational division or second anaphase. Only one division per cycle, that is one cytoplasmic division per sequential division is done. Similarly, only one revision cycle. Okay. Only in meiosis what happens? Two divisions per cycle, that is two cytoplasmic divisions, one reduction division and equational division. Whereas in mitosis, chromosome fail to synapse, no chiasmata formation. In meiosis, chromosomes synapse and form chiasmata. In mitosis, genetic exchange between homologous chromosomes does not occur. In meiosis, genetic exchange through chiasmata occurs between homologous chromosomes. Next is In mitosis, genetic contents of daughter cells are identical. In meiosis, genetic contents of the daughter cells are different. Next, in mitosis, chromosome number of daughter cell is the same as that of mother cell. In meiosis, chromosome number of daughter cell is half of that of mother cell. Daughter cells that are capable of undergoing additional mitotic division. Okay, daughter cells are capable of undergoing additional mitotic division. Whereas in meiosis, what happens? Daughter cells are not capable of undergoing another meiotic division, although they may undergo meiotic division. Then mitosis starts at the zygotic stage and continues through the life or the organism, whereas meiosis occurs in the zygote of algae and fungi. Okay, so all this were the distinguished point between mitosis and meiosis. Out of this 10 points, you need to study at least 6 points for your exam purpose point of view. Okay, at least 6 points you should know it, then you can write. Okay, so just remember this. So here we have completed this chapter, that is chapter number 7, cell division. Thank you, students. Next lecture we will start a new chapter. We will start with a new chapter. Okay, we have completed this chapter today, so we will stop here. Thank you.